This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 84 for May 25th, 2010. Hi everybody, I'm Vincent Racaniello, and today we have a special TWiV from North Central Florida, where my colleague Rich Condit is with me. Hey, Rich.
Hello, Vince. This is really pretty strange, I must say, because usually I'm talking to you, I'm looking at a computer. Now I'm looking at you, and I'm going to have a little trouble adjusting to this.
Yeah, this is an unusual trip. We're in a room at the University of Florida, Gainesville. We have a little audience, a small but enthusiastic audience, right? Can everyone say that? All right. And this is a great place for virology. And I'm visiting to learn about that, and I thought while I was here, we would have a TWiV with some of the virologists here. Of course, this is where Rich is, this is where he works, and whenever I say North Central Florida, this is what I'm talking about. So today we have a special episode where we're going to talk with two professors here at the Department of Molecular Genetics and Microbiology. And first we have Professor David Bloom. Welcome to TWiV.
Oh, thanks, Vince. It's great to be here.
And we also have Professor Grant McFadden.
Hey, Vince. How you doing?
Thank you guys for doing this.
Oh, my pleasure.
It. We're going to talk about what you do today. Grant, with your work on poxviruses, and David, your work on herpesviruses. What do you think of that? Is that a good plan?
Good plan. Good plan.
But before we talk about your science, what I would really like to know, and we can start with you, is how you got to be a scientist.
Okay. Well, I actually, I think, got interested in science very early on. I was always interested in insects and, in fact, collected insects as a kid. But I think what really kind of propelled me into what I'm doing right now was I had a really great high school science teacher.
I talked about that just the other day. High school science teacher.
Yeah, I mean, he was one of these passionate people who put together these great presentations and all these microbiology experiments, from mold growing in dishes to, basically culturing out organisms. So he put a lot of his own money into it, and that just hooked me. And I knew then, from that moment on, I wanted to be a microbiologist.
Where'd you go to high school?
It was at Wooster High in Reno, Nevada.
So, a survey was done recently of scientists, and they asked, why are you in science? And 90% or more, same answer that you just gave, because a high school teacher inspired me to do that.
Yeah.
Any science in your family?
Nope. I was the first one.
Okay.
So after high school, you went to college. Where'd you go to college?
I went to North Carolina in Chapel Hill, and at that point I was basically just interested in microbiology. I knew I wanted to be in some area of that, and I did some undergraduate research in bacteriology lab, and really got into that, and just continued to sort of explore different areas.
After college, you wanted to do a PhD?
Yep.
You go directly from college into a graduate program? I
did. I went to Vanderbilt, and actually my last undergraduate research experience was in an immunogenetics lab. And I really liked the modern genetics, molecular biology introduction I got there, and I was absolutely sure I wanted to be an immunologist. So the person I went to work for and thought I went to work for in grad school was an immunologist. Rotated through that lab, and it just didn't turn me on. And so I ended up going into another lab at Vanderbilt in virology, and that's where I am now.
Wouldn't be that man over there, Dick Moyer.
I think I recognize him, yeah.
So you got your PhD in Dick's lab?
Yep.
I didn't know that.
Yep.
Hey, Dick.
Hi, Vince.
Great.
So that started out at Vanderbilt and then migrated here, right?
Right.
So you came here as a PhD student?
About halfway through my program, the lab moved, and it was such a great lab, I had to follow it here.
Fabulous.
So Dick took the chairmanship here, and I was one of his hires and overlapped with David for, I don't know, just a few months. You were about leaving when I got
about 3 months, actually.
So at some point in your career, did you decide you wanted to be an academic scientist, or did you know where this was going?
I pretty much once I got to undergrad and saw the life of the academic faculty that taught us, I really liked that. I liked the fact that they seemed to have fun teaching, and the research lab setting to me was like, you could do anything you wanted to. You could, follow problems down certain paths. So that seemed to be the perfect environment for me.
Okay. And so from here, here you were working on poxviruses.
Yes.
Right? And that was a pathogenesis project,
yes.
Right? And then what? Well,
so in Dick's lab, basically, we started doing some collaborative work in pathogenesis, actually back at Vanderbilt. And I kind of really liked the host-pathogen interface that we were exposed to in that collaboration. So that took me to UCLA to do a postdoc with Jack Stevens in the herpesviruses. And he was actually a veterinary pathologist by training, so he had these great animal models for looking at herpes infections. And I knew I wanted to learn that and kind of combine some of the molecular techniques to really get into look at the host pathogen interface.
And then?
Well, then I started my first faculty position at Arizona State in Tempe. Started my lab up working on herpes simplex viruses. Was there for about 3 years, and there was also a job search here at UF. There's a lot of attractive features here because of the gene therapy center and some interest in herpes viruses and as large payload gene therapy vectors, and also the Brain Institute here, because herpes viruses are latent in the nervous system. There's a lot of people here that I was interested in working with to look at that interface.
So we got you back.
You got me back, yeah.
Great.
So who was chairman when you came back?
Dick Moyer was chair when I came back.
So you hired him.
Trained him and hired him.
Very good.
I must have done okay, I guess.
You knew what you were getting, at least.
Yeah.
Okay, it's a great story. Let's find out Grant's story. How did you get interested? Was it a high school teacher for you?
Actually, I was very interested in science in high school. I spent most of my career in Canada, and so I grew up pretty close to Montreal, in a small town south of Montreal. And I was very interested in science in high school, but I would say I wasn't yet converted until I went to university, and I ended up going to McGill University in Montreal, and they had some great teaching in biology and biochemistry. They had a terrific biochemistry program, for example. And it took a couple years, but I gradually got fascinated in biology. And when it came time to deciding what to do in grad school, I ended up working on a bacteriophage called Phyx174, which was very big back in the day as a model system to study viruses and bacteria. And I did my PhD with a guy named Dave Denhart. And
anybody who's done a southern or a northern blot knows who Dave Denhart is.
Yes, and in fact, I think Dave can't stand that.
For those out there in TWiV land who don't know the joke here, there's a solution that's used for a protocol that we use in the laboratory all the time that's called Denhart's solution, that has everything, including the kitchen sink in it.
Were you there when he developed that solution?
No, as a matter of fact, Dave had a long and distinguished career studying Phyx174. He trained with Bob Sinsheimer, who was very, very famous in the early days of bacteriophage science. And so Dave had a really remarkable career as a scientist. But he discovered Denhart's solution by accident when he was at Harvard. They were doing blots and looking for nitrocellulose. You could do DNA hybridization on nitrocellulose blots. But the problem was the background was very high. And so when you did the wash and you did your controls, the counts were too high to get very good signal to noise ratios. And so Dave couldn't figure out what to do, so he simply went into the lab and pulled down every chemical on the shelf, tested them one at a time, found 3 of them reduced the background a little bit, combined them, and lo and behold, that was Denhart's solution. And so he will go down, that afternoon of work will go down as his legacy, probably, in science, and he can't stand it.
Well, Ed Southern has his name on a procedure.
Yep.
Other people do. Some people like it, others don't.
Yeah, no.
Personally, I couldn't stand a Racaniello buffer, so I'm happy that.
I had a friend named Wally Mangle who wanted to have a procedure named after him.
Mangle.
Yeah, I'm going to mangle these cells.
That's a great one. So what after McGill, where did you go?
Okay, so I did my PhD at McGill, and at the time, the chairman of that department was a real virologist by the name of Angus Graham.
Sure.
And Angus had a great impact on my career because he was a wise fellow and knew a lot about sort of the business of science. And he was one of the people that I chatted to about what should I do next. And his advice was to go into eukaryotic biology, away from bacterial biology. And at the time, when I graduated, a fellow by the name of Sam Dales, who was very famous in poxvirus research, had just moved to the University of Western Ontario in London, Ontario. And so several people recommended that if I want to get into eukaryotic virology, is to maybe go to Sam's lab. So I did, and started working on vaccinia virus back in the 1970s, mid-1970s. And around then is when I met Rich and Dick at the very first poxvirus meetings at Cold Spring Harbor, I think in 1977. And I got hooked on poxviruses as models to study Mother Nature, and became totally fascinated with them as tools to query Mother Nature, if you like. And I remain fascinated to this day about how these viruses work and how they operate. And so at the end of my postdoc, when it came time to set up my own lab, I decided I wanted to remain in poxviruses, but I wanted to try something different than vaccinia, because most of the people in poxvirus research were working on vaccinia at that time. And I knew there were a lot of interesting viruses out there. And for a while, I started thinking about setting up my own lab on a virus called molluscum contagiosum, which is a human poxvirus, very interesting, quite a fascinating biology. But its downside is that you couldn't grow that virus in the lab. And so that makes study in the laboratory very difficult. So I did a little more research and came up with these rabbit viruses. There's a family of them. And their biology and their history was so interesting, I decided to study them when I set up my first lab, which happened to be in Edmonton, Alberta, at the University of Alberta, back in the early 1980s.
So you and Rich and Dick were both all working on a virus, and one member of that family was about to be eradicated, or was almost about to be eradicated, right?
Yep.
Did that have any impact on?
When I first set up my laboratory at State University of New York in Buffalo in 1978. That was my whole career, right? I spend, I don't know, however long it takes to get yourself to the point where, yes, now I have my lab, I'm all ready to go, right? And they eradicate the disease, all right? It was declared eradicated. But it actually never concerned me, because my motivation all the time was that this was a model system. I was interested in the virus. I wasn't, I'm sorry, but I wasn't interested in the disease, okay? And I figured it would always be a model system. And actually, I'd give credit to the NIH that they have continued to support this over all this period of time. And there's a lot of good that's come out of it.
Well, the reason I bring it up is because, as you know, polio virus may one day be eradicated. Certainly the number of cases globally have been cut down. And perhaps 10 years ago, all of us who work on polio, and then Bert Flanagan, who's in the audience, also knows this story, we were told you should find some other virus to work on.
Right.
And that had a chilling effect on the polio community. They said, oh, there are other picornaviruses you can work on as well. And I suppose that's what you figured. You would do another pox virus. Well, of course, you weren't working on smallpox anyway.
Yeah, very few people were working on smallpox itself. And for example, Rich and Dick were working on vaccinia, which is the vaccine strain for smallpox. Right. And I wasn't even working on either one. I was working on a rabbit virus that is in the same family, but really biologically very different.
So I think the pox virus field was more resilient in reacting to eradication than the coronavirus field.
Yeah, well, one of the problems, as I understand in the coronaviruses, is that when they eradicate polio, they aren't going to want you to have polio in your laboratory. Right?
That's correct.
And we didn't have that problem because we were all working on
another virus.
None of us were working on smallpox. Okay.
So to finish up your career, your first job was in Edmonton?
That's right.
And then your next one?
I was in Edmonton for 16 years in the biochemistry department, and like anyone setting up a new lab, spent years just trying to cultivate my way in my career and develop a new system, because at the time there was very little work going on in the virus that I'd picked. I had to clone it from scratch and ended up sequencing it and studying the basic biology of the virus. And it took a number of years before I really ended up getting fascinated in the nuts and bolts about how this virus actually operates. And I became fascinated in the subject of virus-encoded molecules that serve as inhibitors or modulators of defense pathways of the host. And that has really propelled my career scientifically, the study of these immune evasion, immune modulation strategies that viruses can use. So I was in Edmonton for 16 years, and then my wife and I got recruited to London, Ontario, to a research institute and a university. The research institute was called the Robarts Research Institute, and at the time it was the largest private research institute in Canada, and was plunked in the middle of a university, the University of Western Ontario, which is the site of a couple of ASV meetings, for example. It's been there twice, I think, in London. And so I became a member of the Robarts Research Institute and a professor at University of Western Ontario. Spent about 10 years there. My wife and I spun off a biotech company there in London called Viron Therapeutics.
Which we've talked about on
Yes.
TWiV, I believe.
Mm-hmm. Yes, you talked about one of the molecules that company is developing as for treating inflammatory diseases. So we were in London for about 10 years, and then we got in 2006, got recruited down here to Gainesville. Dick Moyer, again, had his finger in that pie as well, and led the recruitment of Alex and myself down here to Gainesville.
Good job, Dick.
Yeah, I think Dick can retire now.
We're going to have to get you on a future episode to tell us about your reminiscences about virology. Is that a deal?
It's a deal.
Very good. So, David, your virus is never going to be eradicated, most likely, the viruses you work on.
It's going to be tough.
Herpes viruses, because everyone's infected.
Right.
Many animals are infected. So what's your interest in working on herpes viruses?
Well, the thing that led me to herpes simplex, and sort of representative of the broader group, is the fact that it's evolved the mechanism to set up camp somewhere in the body where it can hang out and be latent and be a reservoir for, perpetuating itself in the environment. And the thing about herpes simplex in particular that I find really interesting, and this is one of these sort of global biological observations, is that it can replicate in every cell type in the body. You can, experimentally inject it IV, it goes everywhere. But that's not what happens during the normal course of the infection. It typically replicates locally on the epithelial surface, but it very quickly gets into the nervous system. And once it's in sensory neurons, it shuts down, and that's where it maintains this latent reservoir. So the obvious thing that I've really been wanting to know the answer to, and I guess if I retire, it'd be really nice to know this answer, and that is, what is it about neurons that the virus sees and senses that allow it to shut down, set up this latent state, and then the opposite side of that is, how does it turn itself back on and reactivate?
So what fraction of the world is infected? You're talking about herpes simplex viruses, right?
Yes, my lab mainly works on herpes simplex virus type 1, which is the oral herpes virus that you typically see associated with cold sores or fever blisters. It's a good model system. Depending on the numbers that you look at, whether you're talking about seropositivity or if you're using more sensitive tests for PCR to detect whether people are infected, it's somewhere between 60 and 90% of the population in the United States has HSV1 latent infection. And the longer you live, the higher that number goes. So by the time you're 60 or 70, it's almost 90%.
So some people periodically have reactivations where they have a fever sore or some other manifestation of virus. I know one of my sons always has that, but others never do.
Right.
And they're probably containing viral genomes, right?
Right. In fact, the 90% of the people who have HSV1 never have a reactivation episode that they're aware of. And that's one of the sort of hot areas right now in herpes virology, is the fact that we're sort of redefining latency a little bit in terms of what we mean by reactivation and what we mean by latency. If 90% of the population have latent virus but don't get cold sores, it's not true that they don't reactivate the virus. You actually reactivate it very frequently. It's shed asymptomatically. The virus has been present in saliva or even in your eyes, and you can very efficiently spread it to other people without even knowing it.
So have studies been done where they will follow individuals for extended periods and sample saliva and look for virus?
Yes, actually, with HSV2, there's a lot more data known. So HSV2 is the genital herpes virus. And Larry Corey's lab at the University of Washington has really done some wonderful studies where he has a large cohort of patients, some of them who reactivate frequently and some of them who don't. And he's done a swab. They basically do swabs several times a day. They've even done biopsies looking at the infection. So yes, what you find is if you use sensitive techniques, people are shedding virus about 2/3 of the time.
So this, as you say, really challenges the definition of latency. I go through this every year when I teach the medical students, right? What is latency? And the dogma for a long time has been that in the latent state you weren't making any virus, but that's getting squishy, right?
Right. Yeah, and in fact, people who looked at animal models of herpes simplex infections, for example, over the years, made this observation, many times that, you would get this, what was called spontaneous reactivation, and you could detect this, but people initially interpreted that, I think, as an artifact. The animal was stressed out, or, but I think what that really is showing us that's just the natural state of things.
However, it is true that the genome in a what we call a latent infection is fundamentally different than the genome in an acute infection and is relatively inactive. Is that a fair statement?
Yes, that's right. And it's chromatinized, and actually the latent genomes are set up in a heterochromatin structure, so it's consistent with repressing the viral genes and helping to maintain that latent state. And the other thing that most people aren't aware of is that if you were to look at the ganglia that has many neurons in it, and many of those neurons have latent genomes in them, even when you have a reactivation episode, only a very, very small number of those neurons are actually firing off at any given time. And so you have this large reservoir of potential genomes that could reactivate, but only a small number of those respond to stress at any time to actually make virus.
So in your mind, is this whole latency thing sort of something that would be selected for? It is advantageous to the virus to be able to do this? Do you think about it that way?
Yeah, absolutely. The thing that blows me away, actually, is the people who look at the evolution of herpes simplex have shown that it actually co-evolved with humans prior to the branching off to Homo sapiens sapiens. So it's over 8 million years of history and evolution, allowing it to set up this really sophisticated life cycle.
So why would the virus bother to have a lytic phase at all? It could just transmit along from person to person?
Well, it's got to be able to turn itself back on. So it's got to make new virions. And so the 80 or so genes, lytic genes the virus has, are the ones it needs to turn itself back on, push back the immune system enough so that it can, make enough virus to efficiently spread, probably. And so that's only. It's really, if you look at it in the host setting, the lytic infection is really a phase that it just uses for propagation. Latency is where it spends most of its time.
And the majority of the time, there's no symptoms associated with it. So
that's correct.
If it weren't for the fact that a few percentage of the population were symptomatic, we wouldn't even know about this, right?
That's correct.
Virus is just cooking along.
That's right.
So do you try and understand what controls entry or exit from latency?
Right now, my lab is mainly looking at entry into latency, and so the repression of the lytic genes, what shuts things down, and is that it's probably controlled by some neuronal proteins, but there's definitely some viral machinery that has to hook into that.
So what kinds of experiments do you do to do that?
Well, what we've been doing a lot of recently is sort of branching off from what Rich was talking about earlier, and that is, what is the structure of the viral genome during latency? And we know that there are nucleosomes that are associated with the viral genome, histones that have certain histone tail modifications that help compress the chromatin into a heterochromatic or transcriptionally non-permissive state. So to study that, we use a lot of a technique called chromatin immunoprecipitation, or CHIP, which basically involves using antibodies that recognize certain histone modifications, and we can pull those chunks of DNA down and determine what parts of the viral genome are associated with a certain type of histone and therefore chromatin structure. We use that technique to try to work our way back from the viral genome to the cell. So if we can figure out what those particular markers are that are on the viral genome, we then can ask the question, well, what cellular proteins put those marks there, and then what brought those cellular proteins to the genome, and then just keep working our way back to sort of the initial event, hopefully.
So when the virions enter a neuron, they immediately go into a latent state? They never replicate lytically?
Well, that's I guess that's not completely worked out. The model that I think most people would agree with right now is that, and in fact, this is backed up by, if you look at the initial events that occur when the virus enters the ganglia. So one point I should bring out here is that currently there's really no good cell culture system to look at establishment of latency. For reasons we don't understand, it's very difficult to take neurons out of an animal, put them in culture, and then infect them with the virus. If you do that, what happens is the virus replicates and kills everything. So all of these experiments have to be done in vivo in animal models. So anyway, what happens when you infect an animal with herpes simplex? The virus travels to the ganglia, goes into a neuron, and then one of two things happens. In some neurons, the virus can productively replicate and then spread up the nervous system to the next neuron beyond that. In other neurons, it appears, based on immunohistochemistry studies, that they are never really permissive for the lytic infection at that time, and they shut the virus down. So you either get a productive pathway or latency, and it looks like that it's established, that decision's made right away.
But we don't know why.
We don't know why.
Is there an animal that recapitulates the establishment of latency?
For herpes simplex type 1, the mouse is a good model to look at establishment of latency, and the rabbit is also a very good model to look at establishment of latency.
Right. So do you use animal models in your lab? Because I remember you went to Jack Stevens to learn about that.
Yes, I do. I use both the mouse and the rabbit in my lab.
So what kinds of questions are you trying to answer with those?
Well, basically, one is trying to figure out what are the cellular proteins that are helping the virus establish the latent infection. So we've been actually looking at proteins that are putting histone modifications on. The other is to look at that cell population question. So why is it that some neurons can support a productive infection, and why is it that some do not? And it turns out that sensory neurons in the ganglia are a heterogeneous population. They're specialized. You have neurons there that are responsible for touch, the sensation you get when you touch something. There are neurons there that are specialized to be able to sense pain. and there are neurons there that can sense things like heat. So there seem to be, from work done in other labs, subpopulations of those neurons that are more permissive for herpes infection than others. And what we'd like to know is, well, are there different proteins in those particular neurons that help set up establishment of latency?
So let me ask, if you had to explain to Joe the plumber why his tax dollars should support this, I mean, which you have to do periodically, right?
Right. Not Joe the
What
plumber.
would you tell him? Because we've said that this is,
He's listening.
you know,
He's listening.
oh,
You know.
yeah, I know. Joe, welcome to TWiV. All right. So, what do you say? Can this be a devastating infection under some circumstances?
Absolutely. People who have reactivations with HSV1, the 10% that have the problem, will call you, will email you, and tell you that this has changed their life. HSV2, the genital form of the virus, is even more significant with respect to that. Basically, people say they have psychological trauma from this. I mean, it obviously changes the way that you live, and it can be quite painful. Aside from that, there are some very severe complications of these viruses. What a lot of people aren't aware of is that HSV1 is the leading cause of infectious blindness in the United States. About 50,000 people a year get what's called herpes stromal keratitis, which is an inflammatory disease of the cornea that eventually can result in corneal scarring and blindness. And so, obviously, if you've got this virus, cooking away in your eye and causing blindness, that's a serious issue. So, the way I explain it is to Joe the plumber, would be basically if we can understand how the virus sets up shop and goes latent, we may be able to intervene and block that process.
Great.
Any questions from the audience for David Blue? You're welcome to weigh in. Everybody's laughing.
I'm his student, so I'm tempted to say something snide, but I won't.
Okay. Anything else from you?
Yeah?
All right.
Good.
I just might add that one of our distinguished faculty members in the Department of Surgery had a problem with a herpes infection of the eye, just the problem we talked about. Ended up having a corneal transplant because of that.
So you study this problem for basic reasons, to understand essential questions about viruses, but also maybe someday it can help people with these kinds of infections.
Right.
Is there any, my last question for now anyways, is there any way that one could think about removing all the viral genomes from all of us someday?
That would sort of be the holy grail. I mean, it's, we've all thought about it, but it's a very difficult problem to kind of get at. Just getting access to those neurons, alone would be a difficult problem.
But never give up.
We never give up.
All right. Let's talk a little with you, Grant. Different virus. And so you mentioned you began your career working on myxoma viruses, correct? What does myxoma mean?
Myxoma virus, the name came from the discovery of what this virus did in European rabbits. So the virus was discovered by accident, actually, in the late 1900s, when an Italian researcher by the name of Sanarelli brought some rabbits over to a field station in Montevideo, Uruguay. So he brought these European rabbits from Europe over to Uruguay, and he was a scientist, and he was using the rabbits for something totally different. But he housed the rabbits outside of his station, and within a very short period of time after they were outside, all the rabbits got sick, came down with a new disease that killed every single rabbit. And at the time, he couldn't figure out what had happened, and he went into the veterinary literature to try and find out what had killed all of his rabbits. And he couldn't find anything in the literature that explained this new disease. So he was quite fascinated and ended up collaborating with a number of Brazilian microbiologists to go looking for this, whatever it was that had killed all the rabbits. And it took them several decades of work to identify the pathogen. It turned out to be an infectious disease agent, a virus, that resided in the Brazilian rainforest. And in the Brazilian rainforest, this virus lived in an animal called the tapete. And in the tapete, the virus causes no disease whatsoever. But when a mosquito bites a tapete that's infected with this virus and transmits it to a European rabbit, all of a sudden the European rabbit comes down with a new disease that we now call myxomatosis. So the name myxomavirus came from the description of the lesions, which were very mucinous and looked sort of tumor-like. And so myxoma was sort of the name for a mucilaginous tumor-like lesion that was a manifestation of the disease. So it was a bit of a curiosity for many years, and was, I would say, fairly obscure observation for many years, until the Australians came up with an unusual use for this virus. Many people know that one of the things that has plagued Australia has been incursion by foreign species of different kinds. And one of those incursions was the European rabbit. And in particular, the European rabbit has gotten feral into Australia, and they have proliferated out of control. Anyone who's been in the outback of Australia knows how many rabbits there are hopping around Australia. And the situation got so bad that the Australian government went looking for ways to control rabbit populations. And one of the ways that they elected to do that was to go into a kind of biological warfare with the rabbit. They went looking for a pathogen that would infect and kill only European rabbits, but nothing else, obviously. And so that's what led them to this virus, myxomavirus, because it had been studied and it had been shown that it was the disease was utterly specific to the European rabbit. You could take this virus and inject it into any other animal and it would simply vanish. It would not take hold in any animal other than a lagomorph, and it would only cause disease in the European rabbit.
So this virus is only in Brazil, or is it elsewhere as well?
There are a couple strains. The strain that the Australians used to try and wipe out their rabbit population came from South America. It's called strain Lausanne. But there are also strains in Central America and actually in California as well. And there's a California version of myxoma that lives in the brush rabbit, which you can actually see hopping around in California, for example. And some of those brush rabbits have another version of this virus in them. But to those rabbits, it's not a pathogen. It just causes a little tiny bump. But if you're a European rabbit, then this virus is an extreme pathogen.
So the virus is not in Europe naturally.
Correct.
But by serendipity, when you infect a European rabbit, it's lethal.
Right. And in fact, viruses sometimes do this when they leap from one species to another.
Leap. Is that a...
Sure. Yeah. Hop, leap. But we can never really predict what happens when a virus finds a new species. I mean, look at the case of HIV in humans. No one could have predicted that monkey virus, that is fairly subclinical in monkeys, would turn out to be such a profound pathogen of humans. And this happens, SARS is another example. It's a coronavirus that in its native species is not really a pathogen, but when it mutates a little bit and hops into humans, it can be all of a sudden an extreme pathogen.
Ebola, same thing, right?
Yeah. Yeah.
So myxoma, I take it, does not infect humans.
Well, we know this because as part and parcel of the attempted eradication program in Australia, there were 3 investigators who were leading the idea of releasing this virus into the European rabbit population. There was Frank Fenner, who actually wrote a book on this called Myxomatosis, which is a fantastic book to read, and I recommend it to anyone who's interested in the history of virology to find this book. It's a beautiful rendition of how the Australians tried to use this virus for an eradication purpose.
It's still in press.
Yeah, you can get it.
Print.
And it's a great read. You can read it. It's fresh today. Frank Fenner was a fantastic writer, a very clear thinker. But at any rate, Frank and two other investigators, Clooney's Ross, who was the head of one of the organizations that was promoting this release, and McFarlane Burnett, who went on to win a Nobel Prize, injected themselves with live virus to show that it was not a pathogen for humans. And they were right. And in fact, they injected this virus into all kinds of vertebrate species to show that it was harmless for anything but a European rabbit.
And Frank is still around, pushing 100.
Frank is, I think, 96 and still as lucid as ever.
Maybe I'll inject myself.
So, at any rate, that story, the attempted eradication of the European rabbit, is one of the things that got me fascinated in studying myxoma as a biologic entity. And in the early days, I was most interested in why it was such an extreme pathogen in rabbits. And later, as we started to study it, I became more and more interested into why it was rabbit-specific. And we ended up studying an offshoot of that we didn't predict at all. Namely, could we infect something other than rabbits with this virus? And we ended up learning that we could infect human cancer cells, infect and kill them.
So this was just a random search of other susceptible cells, or you had some goal in mind?
It wasn't totally random. For years, my lab had been studying why this virus was so effective at subverting the immune system. And most of our work was done in rabbits because that's the host where it causes disease. And we ended up knocking out virus genes one at a time and studying how those gene products interacted with the host in various ways. And I don't know if we've mentioned in the past, but herpes and pox are both large double-stranded DNA viruses. And pox viruses contain, anywhere from 150 to 250 gene products, so that ballpark. And of them, about half are probably devoted to interacting with the host, or host modulation, or protecting the virus from attack by the immune system, for example, of the host. And what that means is you can knock those genes out, and the virus is still alive. It's still viable in tissue culture, and you can grow it. And we call them knockout viruses because they've got a targeted gene that has been deleted. And we can study them both in vitro, in culture, and in a rabbit, and ask, has the virus been compromised in some fashion by losing this gene product? So we were doing variations of these experiments for a number of years. At the time, we were paying more attention to how these gene products interact with the host. And often we would look at the target or the pathway that this virus gene product was intersecting with. And it was always fascinating. And I'm continually amazed at how sophisticated viruses can be and how they interact with the host. And if anything, it's a reminder that all of these viruses have clearly co-evolved with hosts and eukaryotic organisms. It's the only way to explain why they know the vertebrate immune system and the vertebrate pathways so well and so strategically. And I'm just fascinated by those particular interactions. But at any rate, so we'd been studying these knockout viruses for many years in terms of how they intersect with the host. And we gradually discovered that some of the knockouts also had phenotypes that we didn't predict based on what we'd thought about in terms of what their targets might be. And one of the phenotypes that was really fascinating is something called host range phenotype, which means that when you knock that gene out of the virus, even though you can grow it in tissue culture, has some kind of defect. It won't grow in a cell type or a tissue type that the parental virus can grow in. So in other words, it suffers a defect in how it can recognize and grow in certain kinds of cells or certain kinds of tissues. And although we could never predict it, whenever we stumbled upon a host range phenotype, we would learn something about what the virus uses to discriminate in the host cell in terms of whether or not it's going to replicate or be non-permissive. And while we were doing those experiments, we began to realize that the virus had very specific molecules that it used to help it replicate in some cells or some tissues compared to others. And this got us and the people in our lab interested in this business of tropism determinants in the virus, what it is that they're doing, and how does the virus select what kind of cell is going to be permissive or non-permissive. And as we were doing these experiments, we began to stumble upon something that I know is familiar to you, Vince, and many virologists, which is when a virus infects a mammalian cell, it's not simply a case of the cell passively sitting by and waiting to be wiped out by a virus. In fact, when a virus touches a mammalian cell, there's a very rapid dynamic response from the cell, often in an attempt to protect itself from the infection. And there are many different ways that mammalian cells try to innately protect themselves from an infection. And we started studying some of these pathways, and we stumbled upon the observation that, for example, there are very specific pathways that protect cells from infection by this virus, and probably all viruses. And one of those pathways is the type 1 interferon response pathway, which is a very famous and well-known response pathway that cells can stimulate to protect themselves transiently from virus infections. And what we learned is we could take this rabbit-specific pathogen, and if we put it, for example, on primary mouse cells, we know that mice are non-permissive. We know that primary mouse cells are also non-permissive for the virus replication. So if you take the virus, you put it on these cells, the virus vanishes. In essence, a tug of war has happened between the virus and the host cells have won that particular tug of war. But we stumbled upon the fact that if you knock out the interferon response pathway of those cells, all of a sudden these non-permissive mouse cells behaved phenotypically just like rabbit cells. They became permissive for the infection and the productive infection of this virus. So that got us to thinking that maybe the determinants of tropism on the cell side was entwined with the innate signaling pathways, the capacity of the mammalian cells to generate responses that determined whether or not a given infection was going to be permissive or non-permissive. And this was part and parcel of the reason we started to ask whether or not the virus could infect cells outside the rabbit that maybe had defects in these pathways. And this is what really led us on to the question of whether or not the virus might infect cancer cells. Because cancer cells are well known for being defective in many kinds of signaling pathways. Because in many ways, cancer is a genetic disease, and in large part, it's a genetic signaling disease of individual cells. Their signaling circuitries goes haywire. And I remember I was giving a seminar at the University of Ottawa. This is about 8 or 9 years ago. And a fellow in the audience by the name of John Bell came up to me, and John said after the seminar, do you realize, all these different signaling defects that are in cancer cells, Have you ever thought of checking to see if your rabbit virus might replicate in human cancer cells? And so we received a whole bunch of cancer cells from something called the NCI 60 collection of cancer cells, and which is really 60 different cell lines that have been developed from different kinds of cancers from patients in various classes of cancer. And it's sort of a panel, a spectrum of the kinds of cancer cells that can exist out there. And we began to test whether or not this rabbit virus could replicate in any of them. And lo and behold, about 2/3 of them were permissive for the replication of this virus.
Whereas the normal counterparts of those cells were resistant.
Well, that's another interesting aspect. It turns out that if you take primary human fibroblasts that are as fresh as you can get them and put myxoma virus just on those cells are permissive, even though you wouldn't think that they would be. But a postdoc in my lab, Eric Barty, has shown recently that if you put interferon and another cytokine called TNF, tumor necrosis factor, on those primary cells, it makes them completely resistant to infection by myxoma. So it turns out there are certain cell types that respond to myxoma infection by making interferon and TNF. Cells like macrophage, for example, they make TNF and interferon when you infect them with a myxoma. So if you take fibroblasts and macrophage and mix them, the mixed population, infect them with myxoma, interferon and TNF is produced and it protects all the cells from infection. So mixed tissues that, for that reason, we think are completely resistant to the infection of this virus. But cancer cells, for reasons that I don't completely understand, and I don't know if it's really well understood at all, are capable of sometimes ridiculous amounts of growth and replication and metastasis, but as a consequence of that, they sometimes pay a price. And one of those prices is to compromise their status, their intracellular milieu, by losing some of their capacities to induce certain pathways. And some of those lost pathways include antiviral signaling pathways. And so I think it's a general phenomenon that cancer cells often become more sensitive. One of their Achilles heels is that they become more sensitive to the replication of a number of viruses by virtue of losing some of their antiviral signaling pathways. And I think this is part and parcel of the reason oncolytic virotherapy, which is using live viruses to infect and kill cancer cells, is probably based on some of these genetic defects in cancer cells themselves. And that's what led us on into investigating myxoma as an oncolytic virotherapeutic.
So what kind of cancer have you looked at specifically with myxoma?
So far, our first collaborations in this area was with a scientist and a neurologist from the University of Calgary, a fellow by the name of Peter Forsyth. He studies brain cancers of various kinds, gliomas, glioblastomas, medulloblastomas, etc. And Peter has told me that so many of these cancers are intractable, and many of his patients are young kids and young adults, and the prognosis for many of these cancers is very poor. And Peter says he needs a reason to get up in the morning, and the reason is research. He is always looking, and people like him are always looking for new therapeutics, new ways to treat brain cancer. And he heard about our results and offered to set up a collaboration to see if we could use myxoma virus to treat gliomas. And we published that paper a number of years ago now, and it worked exceedingly well. We could take human gliomas that were growing in, that were transplanted into the brains of highly immunodeficient mice. So these are called xenografts. So you take human cells and engraft them into another species, in this case mice. If those gliomas are left to their own device, they'll kill the mouse within a couple weeks after the transplant, sort of mimicking the malignant qualities that happen in actual brain cancers. So we gave Peter some of our virus that was tagged with green fluorescent protein, so-called GFP-tagged myxoma, so that we could follow the replication of the virus by virtue of the generation of green fluorescence. And when Peter injected this virus into the gliomas, it was really pretty amazing. The virus percolated through the tumor tissue very much the way it percolates through rabbit tissue. And in many ways, I sometimes think of oncolytic viral therapy as a kind of tissue-specific pathogenesis. It just so happens the sensitive tissue is cancer tissue, but the fundamentals of the pathogenesis are probably very similar between a susceptible, let's say, rabbit tissue and a human cancer tissue. And I'm really quite fascinated by the similarities and differences between those two environments. But the bottom line is that we cured essentially all of those mice of their gliomas, and that is what the initial study that convinced me that there's something to this. And
has that been put into people yet?
A number of oncolytic viruses, of course, have been put into people. Oncolytic viral therapy as a discipline has been around for a number of decades. And in fact, there's if any of your listeners are interested in the subject, Steve Russell wrote a terrific little review of the history of oncolytic viral therapy a couple years ago. We can put a link up on your site if you like. But the idea of using live virus to infect and kill cancer cells has been around for maybe 100 years. In the early days, it was anecdotal that certain viral infections would help treat or help some cancer patients to regress their cancers. So the bottom line is that there are many different viruses that are being explored for oncolytic viral therapy. And the field of oncolytic viral therapy meets every 2 years. There's an oncolytic virus meeting. The next one is in Las Vegas next year. And in fact, when you're at the ASV this summer in July, there's a satellite symposium just before ASV begins on the Saturday morning before the main core meeting begins. Terry Dermody has set up a satellite meeting on viruses and cancer. And if you're there on Saturday, you can come and sort of listen to the stories. But I became convinced that as we were studying this rabbit-specific pathogen, that in fact it could also be a very potent pathogen against cancer tissues in a very tissue-specific fashion. And so since those collaborations, we've done a number of things in our own lab. We've studied metastatic melanoma, for example. With Peter, we've looked at medulloblastomas and rhabdoid cancers in the brain. And now that I've moved to the University of Florida, we've started to investigate whether or not we could use this virus to selectively eliminate human cancer cells that contaminate bone marrow grafts for treating of cancers using what's called autologous bone or blood hematopoietic stem cell transplantation.
So with any of these oncolytic viruses and the trials that are going on, are there any real successes that come to mind, or are we too early in this?
Well, let me put it this way. One of these viruses that was developed in the earlier days of oncolytic viral therapy, which is an adenovirus, has been approved for human use. It's been licensed in China. It's not yet been approved in the U.S., but it is a mutant of a human adenovirus that has a particular predilection to replicate better in cancer cells because of the way that the knockout interacts with the signaling pathways of cancer cells. So it has only fairly recently been licensed, and I would say it's still too early to know if it is the ideal oncolytic. It's been approved for head and neck cancers in China. Elsewhere, a number of other viruses are in various kinds of clinical trials, various stages of clinical trials. And, for example, Newcastle disease virus has been in trial. There's a vaccinia virus variant that a company in San Francisco is developing for liver cancer right now. Each of these oncolytic virus meetings, we hear a little bit more about what's going on with the different trials and the different stages. My own opinion is that oncolytic virotherapy is still in the very early stages of development. Really, the ability to genetically manipulate virus has only been around for a couple decades. And the field, and we're a part of that field, is still learning about how best to use viruses. I think in the long run, oncolytic viruses are going to find their way into clinical practice and into clinical oncology once we learn strategically how to use them.
Well, as you've pointed out, the interactions that you're talking about have to do with the virus interacting with cellular signaling pathways. And hardly anything could be more complex than that. So it's going to take a while to sort this out.
Correct. And in fact, most people believe that the different cancers, in the early days, a lot was thought about individual oncogenes or individual tumor suppressors, whereas now I think there's an appreciation, now that there's this mega sequencing of different kinds of cancers, is that cancer is really pathway diseases. And the next stages of therapeutics needs to have strategies that go after dysregulated pathways as much as individual targets. Right. And I think one of the strengths of oncolytic viruses is that viruses can tackle pathways in mammalian cells very strategically. And so I think oncolytic viruses are going to find their way into clinical practice, but there's still a lot of work to be done in terms of how to use them specifically. In our case with myxoma, we don't have yet clinical grade of the virus because you have to go through very specific manufacturing techniques to make something like a virus ready for a patient trial. But I'm sufficiently emboldened by where we are right, what I see in terms of the data, that I can foresee in the not too distant future us having the capacity to make clinical grade virus and enter into human trial.
So you said in the long term you think it will be useful.
Yes.
What's long term? 10, 20, 30 years?
Well, you know, the road is littered with people who make predictions about where medical research is going and how long it'll take to get there. I would say I don't know what the long term will be, but I certainly expect to see in my lifetime specific uses of oncolytic viruses for certain cancers. And as clinicians learn how to use them and scientists get more strategic in showing how best to use them, that their uses will grow at some point in the future.
And since you're regularly exposed to myxoma virus, your lifetime could be pretty long.
Well, Frank Fenner would agree.
Well, you know, Jonas Salk started working on a polio vaccine in 1950, and he's told everyone, this is going to take a long time, don't make me give you numbers, and then 4 years later, he was done.
Right.
So sometimes you're surprised. Scientists are always circumspect, but the public wants to know, is this going to happen in my lifetime or another life.
Yeah, and we all ask ourselves the same questions. Most of us also in the research community would like to see something good happen from what we do.
Sure.
And so I'm motivated by the fact that I think there's a real likelihood a number of these viruses are going to find their way for actual treatment of real cancers in humans. And I'd like to think it's in my lifetime.
Well, we will periodically get in touch with you and find out what's going on.
Right.
How's that?
Yeah, no, I find the field of using viruses to infect cancer cells really fascinating and interesting. And not only is there going to be practical implications, but I think we're going to stumble upon interdigitations between viruses and mammalian cells that we don't suspect right now.
Sure. Well, as you'll see, we have an email from a listener who is interested in this, and many of them are. But as always with virology, you always discover new things about cells. That's the expectation.
What I love about viruses is that they're a gateway to Mother Nature.
So let's do some email, but before we do that, we need a word from our sponsor, which is Data Robotics, and I'm very happy to tell everyone here in the audience about this product. This is a backup solution for your computer. It's called the Drobo. It's a wonderful little black box, and it contains slots where you simply slide in hard drives, you hook it up to your computer, and you copy your data to it. Anybody here have a Drobo? So for the record, you have one? One person. You're going to get one.
I
Excellent.
really want one for my house.
Well, you should enter the contest. You could win one.
I actually did it on my BlackBerry while you were setting up.
All right. So these are wonderful solutions. They come in 4-drive, 5-drive, and 8-drive sizes. They can be networked, so you can plug them into your Ethernet network and have access to everyone, or you can just put them in your ports on your computer. And if any one drive fails, you simply pull it out and put a new one in, and all the data are restored on the drive. So these are fabulous solutions that are used in many, many industries, and we think for science this is a great idea. In my lab, I have all my data on a Drobo. I have several Drobos at home, and they are wonderful products. And I'm really happy that the makers of Drobo, Data Robotics, have decided to support This Week in Virology. They're particularly interested in getting into the science and research field. They think there's a real need for backup, and as you know, we're generating more and more digital data nowadays, so this is a perfect solution. So Data Robotics, or Drobo, has made a nice landing page for TWiV, and since we're live here, we can check it out, drobo.com/TWiV. I think that's it. Here we go. Welcome, TWiV listeners. So you can go here and do these data storage headaches sound familiar? Drives crash and I lose data, time, and money. So you can find out all about all the different models of Drobo here. You can see here's one of the 4-drive models, and here's an 8-drive model down here if you have lots of data. So right now a 2-terabyte hard drive is about $100. And you can put 4 of them into here and have 8 terabytes of storage right away. And eventually they'll be up to who knows how many terabytes. So it's a wonderful solution.
Having spent a little time with you, Vincent, I can see that you could fill one of those up pretty easily.
I can fill it up very easily, absolutely. I have all these podcasts take up a lot of room. All my lectures, which I video, I keep on the Drobo. And I don't want to lose any of this. And of course, I put all my students' theses, all of our data go on it as well. It's a fabulous solution. So if you go to drobo.com/TWiV, you can use the product code TWIVPod, and you'll get $75 to $500 off a Drobo. Or you can enter the contest, and the link is bit.ly/TWiVDrobo, and that's also on TWiV.tv, so you can find the link to that. The contest this time is the third Drobo we're giving away. It is simply a multiple choice questionnaire where they'd like to find out what podcasters would like in a backup solution. So as soon as you enter fill out the questionnaire, you'll be entered in the contest, and then in about a week there'll be a random drawing. So it's very easy, and you have a really good shot of winning this. There are not a lot of entries, so keep your fingers crossed. The second contest was actually an essay contest to answer a question. Could influenza virus be engineered to replicate in bacteria? So we thought that would be a fun question. We got lots of good answers, and Rich and I graded them because that's what we do. Right? We're good at grading. And
Did you guys publish the winning essay?
no, we didn't publish it. We should publish.
That's a good
the winning
idea.
essay.
That's a good idea.
I'd love to read it.
Yeah, people have asked for that, and we should publish some of the runners-up were excellent, and
There was a couple of really flaky ones that were deliberately flaky, and they were very entertaining as well. I really appreciate it.
I think it's great that people entered and did this, and many of these people were not scientists at all. They're IT people or in other fields who just wanted to give it a shot. So it's fun. So we really are grateful to Data Robotics for their support of TWiV. Thanks. Okay, let's do a few emails before we wrap this up. The first one is from Matthew, who writes, "First, love the podcast. I listen to it on my way to work. Don't change a thing. I recently wrote to you asking about the possible link between a high arginine diet and herpes simplex outbreaks in humans," which I believe Rich consulted with David, and we ended up concluding that there was no benefit to having a high arginine diet in terms of herpes. While researching, I found this study, which to my untrained eye seems to be saying that HSV1 DNA in the neuron nucleus is histone bound and folded into chromatin when it is latent. Sounds familiar, I think.
Right up your alley, David.
This suggests an epigenetic control pathway via HDAC inhibitors. Perhaps diet could play a role in suppression of HSV1, as diet is linked to epigenetic silencing of genes in animal DNA. Please, could you talk a little about the epigenetics of viruses? Well, here's the paper. It's a paper by Neil DeLuca, Epigenetic Modulation of Gene Expression from Quiescent Herpes Simplex Virus Genomes. Maybe David can shed some light on this.
Yeah, no, this is a great paper, and actually an area of a lot of interest right now in herpes simplex virus biology, as well as some of the other persistent viruses too. But yes, these viruses are packaged in nucleosomes storing latency, and there's been a lot of interest in looking at what kind of regulatory events happen at the chromatin. So one of the things that's kind of interesting in terms of how viruses have evolved maybe to shut down their genes and maintain latent is the fact that they have to get around some host defense mechanisms. So there's a lot of evidence that suggests that when just naked DNA comes into a cell, that the cell senses that and tries to shut it down. And one mechanism for doing that is by putting on histones and ultimately by methylating the DNA. Then that's sort of just some innate types of responses. What it looks like has been happening from work out of some of the labs in the herpes virus field is that HSV sort of tries to put a little twist on that. It doesn't necessarily mind having some help in shutting down its lytic genes if it's going to set up this latent infection, but it wants to do it in a way that allows the virus to still reactivate from latency, so sort of on its own terms. And so a lot of the work that's being directed towards understanding how the latent genomes are chromatinized are looking at what flavors of chromatin, how tightly repressed are these markers.
That's what you were talking about by doing CHIP to identify various histone proteins
right
associated with the genome. So Matthew had asked, could diet play a role as diet is linked to epigenetic silencing? I don't know anything about that. Do you know?
There's been a lot of work being looked at sort of environmental factors that basically drive different pathways of epigenetic modifications on cellular genes. I don't know if some of you probably have seen in Science recently, there was a paper looking at twins, and they have this identical DNA sequences, but what they found out is that their epigenetic profiles can be very different depending on what they eat, where they live, presumably. So this definitely could plug into how viruses could reactivate. So yeah, I think, there's a lot of people who insist, I think as you mentioned on the program a couple of weeks ago, that changing their diet has affected how frequently the virus reactivates. So it's known that diet, stress, environmental stresses, sunlight, all change the epigenetic profiles, transiently usually, but it could definitely play a role.
So it's an idea at this point. Squishy, but an idea.
It's an idea.
You like that word, squishy?
I like the word squishy, yeah.
Well, we concluded a few weeks ago that a high-protein diet would be good, It might be good. And if you want a high protein diet, you just high arginine diet. Sorry, if you want a high arginine diet, you just eat a lot of protein. You eat a lot of meat or tofu or beer, because beer has a lot of protein in it. Right, there you go. Okay, great. Thanks, Matt. Jesper writes, Dear professors, a while back you discussed the idea of applying mutations to computer viruses to see how they evolved. Someone wrote in and explained that it simply doesn't work that way in machine code. So we asked someone, could a computer virus evolve? And a very interesting IT person wrote back saying, code is too strict. It can't vary. You break the program if it changes. So here's another IT person responding. I am currently reading The Red Queen by Matt Ridley. Great book, I must say, on the discussion of sex as a means to out-evolve the threat from parasites. He mentioned a computer program named Tierra. Tierra is a virtual environment where programs compete for resources, mainly by copying themselves, and thus their species would occupy more of the environment. The author of Tierra threw in a small randomization in the copying algorithm to simulate mutations. As in the biological world, most mutations were detrimental and caused program malfunctioning and thus death. But somewhat to his surprise, some mutations appeared that didn't kill the host. First, they had no impact at all. Then suddenly parasitic programs came into being. These programs had no code for reproduction, but used the required instructions from other programs. Some of the initial mutated programs were immune to the parasites, and on it went. So he gives us some links to these to this project, which we'll put in our show notes. That's really interesting that you could actually mimic evolution.
Yeah, I just skimmed this first link, and it's I didn't get to it in detail, but it looks like a pretty easy read and really very, very interesting.
Sounds very biologic, doesn't it?
Very biologic and very viral, actually.
Yeah.
It's this guy got surprises when he messed around with the code. Stuff came up he didn't expect at all.
I think this is very interesting to try and simulate viral evolution. So it's great, Jesper. All the best. I thoroughly enjoy TWiV and TWiP. In terms of podcast competitiveness for attention resources, you rock. All right. Thanks, Jesper. Jesper is from Sweden, just to give you an example of the reach of TWiV. Jim writes, thought you would like to see this article from the Mother Nature Network. So Jim is an interesting fellow. He writes to us a lot. He's retired. He's a nuclear power plant operator who's retired and loves to learn about science. So he sent us an article, New Handheld HIV Detector Fits in Your iPod Case. And so this is an article from the Mother Nature Network, which I'm not aware of, but here is what looks like an iPod. New device invented by researchers at Palo Alto Research Center, where the mouse was originally invented. Make it easier to detect HIV on the go. It's the size of an iPod, and it basically provides a full immune system checkup in less than 10 minutes. You take a little sample of blood, pinprick, and somehow you put it in this device, and it counts CD4 positive T cells. So basically it gives you a readout. It's a little flow cytometer in your pocket.
I guess.
Right?
We couldn't figure out exactly how it was doing this, okay?
Millipore now has a pipette that has a cell counting system built into the chamber, so I'm assuming it probably uses similar micro-optic technology to just count and delineate between cells. That's fascinating.
Yeah, it says here they're competing with a dozen other groups, and these are funded by the Gates Foundation. So that's great. I have a view that one day, and this is a long time in the future, you're going to get up in the morning and look in the mirror, and the mirror is going to scan you and say, no infections today, haven't they? I really think that's going to happen. I hope it happens in my lifetime. I don't know. All right, we have a letter from Bill who writes, Doctors, I'm still hooked on TWiV, and are you still thinking about a TWiV episode focused on this cool emerging virus-based cancer therapy? And he sent us a link to a JCI paper entitled Anti-Angiogenic Cancer Therapy Combined with Oncolytic Viral Therapy Leads to Regression of, I don't know the rest of tumors in mice. The company is now about to enroll its first patient to a phase 3 registration trial focused on head and neck cancers, but there are many studies being conducted in and around the principal discovery that reovirus exploits the RAS flaw that is common to 70% of all tumor cell lines. It's shaping up to be a huge story. Time for you guys to get on it.
I think we just did.
We're on it. You know anything about this story?
Well, I actually pulled this paper out. It's a recent paper in the Journal of Clinical Investigation, and it's a really interesting story that brings up a very important issue with oncolytic virotherapy in general. And the issue has to do with delivery of the virus. How do you get virus to all the different tumor sites, particularly when they're metastatic or micrometastatic? And it is probably the number one technical issue that needs to be solved in order for oncolytic virotherapy to have really wide applications for many kinds of cancers. So this particular paper is using reovirus. And reovirus, it's a really interesting virus. It's a double-stranded RNA virus. And in nature, it doesn't really cause very much disease at all. And in fact, I think one of the things that people who study reoviruses bemoan is the fact that it doesn't. And so it has a low profile, and there aren't that many labs studying reoviruses. A fellow by the name of Patrick Lee in the 1990s discovered that cells that have a defect in the RAS signaling pathway grew this virus better and to higher titers than those where the RAS pathway was not defective. And so Patrick came up with the idea of using reovirus as an oncolytic candidate. And there is a company called Oncolytics that is dedicated to clinical development of reovirus as a therapeutic. And this paper comes from Richard Weil's group, in which they looked at how could we improve the delivery of virus to tumors. And the strategy they use here is to disrupt the capillaries that service tumor. In their case, they use B16 melanomas. Now, normally, when you inject virus IV, intravenous, the problem with that delivery mechanism is a lot of it gets cleared to the liver, and so only a tiny amount actually makes it to where it's needed in the tumor burden. And it's further complicated by the fact that if the tumor is innervated by capillaries, if the virus can't cross the capillary into the tumor bed, then it just won't get to where it is actually needed. And so in this paper, they came up with a pretty cool strategy in which they pulsed the endothelial walls with a stimulatory protein called VEGF, vascular endothelial growth factor. And they had to do it in a particular way where they starved the endothelial cells of VEGF and then gave it a pulse of synthetic VEGF. And when they did that, it made the capillaries infectable by this virus, and it propagated the virus into the tumor bed and increased its oncolytic capacity. So a lot of strategies are being tried in the business to find ways to better deliver oncolytic viruses to the actual tumor cells. And in fact, one of the reasons that we've decided to explore using viruses to purge cancer cells from bone marrow grafts is we can deliver the virus to every single cancer cell because the bone graft, the marrow graft, is in culture, and we can actually get the virus to every single cell. Whereas in this paper, they're tackling the more difficult problem of how do you get the virus into all these different tumor beds. So they've had, their report here, they can prolong some survival, and the results are very intriguing. And I think it's the kind of thing that the field is doing now to explore. So Vince, you picked up something, figure 5.
Well, I'm just showing Rich a stained cell monolayer. I know he loves that so much. It's not
plaque assays, yeah.
I don't. It's just CPE, I think. with and without VEGF, but I know you like this. I like it too. I think it's great. So there's a Calgary company, so you must have known about.
I know of the company, and I know Patrick Lee as well. Since this time, Patrick's moved on to Dalhousie University in Halifax, and Patrick is still completely interested in real viruses and real virology. And one of the nice things is that this is a case of a virus that it's hard to make an argument of its clinical importance because it doesn't cause disease.
Sure.
And yet doing some fundamental science on it, they came up with these ideas of exploiting the virus in a new and different way by virtue of the discoveries that were made by studying it from fundamental principles. So I really like that.
I think the VEGF idea is great. Sounds wonderful.
Yep, very interesting idea.
So thanks, Bill. And we will do more viral oncotherapy. Maybe at the ASV meeting we'll get some people who are at that meeting. I know. So Terry Dermody is a reovirus person.
Yes, absolutely.
He's gotten interested in oncotherapy with reovirus.
Yeah, he's the one who's organized the satellite symposium on the Saturday of ASV.
All right, let's do two more, and we'll wrap this up. The next one I picked out for you, Dave. It's an equine herpes virus outbreak in New Jersey, my home state. This is a total of 6 New Jersey non-racing horse farms were under quarantine because of an outbreak of equine herpes virus, which doesn't usually cross my radar. You ever think about equine? So there are apparently herpes viruses of lots of other species other than humans.
Yeah, absolutely, and a lot of them are serious veterinary pathogens. And in fact, the equine herpes virus is one I'm familiar with because I had a relationship with a veterinary pharmaceutical company to build a recombinant equine herpes virus vaccine at one point before they were bought out by a larger fish, and then that project died. But it's a very serious problem in the horse industry.
Yeah, it says here that 2 of the horses died. And I suppose if it's in a racing horse, it's very serious because they're worth lots of money. One thing I noticed here, so these establish latent infections at birth, I assume, and then the horses are periodically producing virus? Is that the idea?
Yeah, at birth or shortly after birth is a typical time where the horses get these infections. It's serious, particularly if mares get them while they're pregnant, because certain strains or certain epidemics of this, particularly EHV1, can lead to what they call abortion storms, where it just devastates horse farms.
So they're ubiquitous in horse populations, and you get an acute febrile respiratory disease on primary infection, and then the latent reactivation.
Big deal in Florida. There's a big horse industry here.
Yes, that's right.
The virus is in Florida as well?
Yes, they are. In fact, it's hard to find horses that haven't been exposed to these after they get to a certain age.
I noticed as part of this article that apparently these undergo a fair amount of antigenic variation, so that the vaccines, it's hard to keep up with them. Is that right?
Yeah, that's a large part of it, and that's also that they're. So the history behind these two is they're both alpha herpes viruses, and for a long time they thought they were just one virus. But eventually they separated them out into 2 different types, EHV1 and EHV4. So EHV1 is, I think, the one that's definitely feared more. It's it's often called equine abortion virus. That's the one that's associated most often with abortion storms. EHV4 is associated almost always with just this respiratory disease, and so it's equine rhinopneumonitis virus is what another name for that one are. But there's overlap. They both cause the respiratory symptoms, but the pathology suggests that EHV1 has a greater chance to cause viremia and spread throughout the animal, and that's obviously what's associated with the abortions.
So this is interesting. The vaccine story, parenterally administered live vaccines are licensed in some countries but banned in others. And an activated vaccine is the only product currently recommended as an aid in prevention of abortion. And immunity lasts for only 2 to 4 months. You have to get boosters every 3 to 6 months. So it looks like we need a better vaccine.
Yep. And that's why there was a push for the project I was involved with to build a recombinant vaccine that would be safer. But the problem is the expense of building recombinant vaccines, particularly for veterinary use, is very high.
So this report, which was sent to us by Len is another faithful listener. This is from ProMed Mail, and if you don't know about ProMed Mail, it's a reporting service of the International Society for Infectious Diseases, daily reports on outbreaks all over the world. Terrific reading to find out what's going on. So thanks for that, Len. And the last one is a joke from Gary, who is at the Armed Forces Institute of Pathology. So the background to this is I made up a joke a couple of weeks ago. 2 viruses walk into a bar. Bartender says, "Are you old enough to drink?" And one of the viruses says, "Yeah, I'm positive."
Ha ha ha. Good.
Bert would get that. So Gary writes, the original version of the I'm positive joke, as I heard it, is an atomic physics joke. 2 atoms are talking, and one says to the other, "I think I lost an electron. Are you sure?" "I'm positive." Thanks for the excellent podcast. All right, thanks, Gary. All right, so we have lots of emails, as you can see here, but not enough time to get to them, so we'll do it next time. And let's quickly go through a couple of science picks of the week. You can go first, Rich.
Okay, so since we're assembled here in Florida, I'm going to do another nature photography site. We'll put the link up. There's a local photographer named Charles Littlewood, and probably a lot of people even here don't know about this guy. I found out about him because we had staying with us in our home for a while a woman who's a volunteer ranger out at Paynes Prairie, which is a local prairie, a wildlife refuge and stuff. And she came back all excited one day because she'd escorted this guy out early morning on the prairie, and she's fairly good friends with him, but they'd had a particularly good day, and she came back and they just posted all these pictures. They just posted all these pictures on his site, and they're just absolutely gorgeous photographs of all the wildlife out on the prairie and locally. Woo! Vince has got up on the screen a big gator. They're beautiful photographs. So, in particular, if you like nature photography, have a look, and if that interests you, come down and have a look at Paynes Prairie, and I'll give you a tour myself.
I never got to see a gator.
Well, we aren't done yet. We're going to go by Lake Alice. On the way out to lunch, we'll see if we can find you a gator.
All right, my pick is from Sophie, who writes, I want to suggest a science-y baking blog. There are, among others, several immune cell cookies and mouse cakes. We all know that mice are a very important animal. No fruit flies, though. It's a really nice way to learn about subjects, and you could make interesting games with them. You choose a cookie upside down, and if you can name the kind of cell, you get to eat it. I know that would make me learn a lot faster. So let's take a look. This is called NotSoHumblePie.com. Let's show you what this looks like. There's some great baking items here, but it's a science-themed baking site. It's called Not So Humble Pie. So you can see some cell cookies here at the top, which are really, so people send in their
Can we get
baking.
a pox cookie?
You could make a pox cookie or a coronavirus cookie. Absolutely. I was thinking of that this morning. Yeah, that would be, so here's a red blood cell in the middle. That's really nice. So this, down below, there are all kinds of interesting cookies. Here's some molecular cookies here. But wait till you see this one. This is great.
This is great.
It's nice to have a live audience.
Yeah. This is a brain cake.
This is a brain cake. Look at this with eyes in the front.
Oh, that's nice.
And, of course, in the back it's damaged and leaking.
That looks delicious.
So would you eat that? That's delicious. So this is molecular biologist. Oh, here's some mouse cookies. This is from molecular biologist Carolyn. So people send in photographs of their cookies. Oh, this is a GFP mouse, the green one. This is a nude mouse. Cover your eyes, kiddos. So anyway, it's a great site. Thanks for that, Sophie. We'll put a link for that in the show notes. Grant, did you want to pick something?
Yeah, I just finished reading a fascinating book called The Strangest Man. It's the story of Paul Dirac, who was one of the fathers of quantum mechanics. And he was a very odd character, but he was brilliant and played a major role in the early days when people were trying to understand the atom. That's it. That's the one. And What prompted me to read this book was a book that Rich Condit got me to read about a year ago called The Making of the Atomic Bomb.
Yeah, which was my first pick.
Which
time
is a
around.
fantastic read. And what I like about it is it talks a little bit about the science behind the making, of the atom and the energy within the atom. And it talked about some of the scientists that were involved in those early days, people like Niels Bohr and Einstein. But one of the people in that book was Paul Dirac, because he and Schrödinger and Heisenberg were sort of involved in breaking away from Einstein and getting into this, our more modern quantum view of the atom. And this book is about one of those three, Paul Dirac, but it talks about the life and the times, and it's just a terrific read. If you like science stories that deal into the peculiarities of scientists, because he was he was odd in a way that, in retrospect, he probably was some kind of autistic or maybe an Asperger's. You get the impression that nowadays he would be classified as something of that order. But he was clearly brilliant, and the book does a great job of trying to explain quantum mechanics and uncertainty and how antimatter and matter came about from just thinking about them. Mm-hmm. And so it's a great read. I highly recommend it.
A lot of comments here on the site from a variety of sources about this book, so apparently very well regarded.
Yeah, you can add my endorsement at the bottom.
Excellent.
I loved it.
Well, that's great. Excellent. Very good. David, did you want to share anything with us?
Yeah, actually, just from a symposium I just came from, there was it's a sort of a new idea that's been put forth by Stan Prusner, and he's written a review that I don't have the link for recently. But basically, as most of you know, he was the guy who won the Nobel Prize for the prion link with disease and the mad cow disease. And what he's now actually gathered a lot of data, and the hypothesis that's being put forward is that a lot of neurodegenerative diseases that involve these types of fibril bundles in the brain, Alzheimer's, Parkinson's disease, they have these common features of these bundles forming, that a lot of these have properties of being catalytic, and that they can be transferred from animal to animal and cause certain diseases. So I think the suggestion is that the prions may be a much broader type of pathogen. They're not viruses, but they're transmitted in an infectious pathway. So that just struck me as being fascinating.
Maybe you can send us the link to that.
I'll do that.
Because I'm not finding it here. But yeah, we on TWiV, we love prions, don't we?
Yeah. Spent quite a bit of time on prions.
All right, great. Thanks, gentlemen. Well, that'll do it for another TWiV. You can find all our show notes and a place to listen to TWiV at twiv.tv. And as always, send us your questions and comments. You can also find us on iTunes, at the Zune Marketplace. And now we're on an application called Stitcher. So if you have a mobile smartphone, an iPhone, an iPod Touch, BlackBerry Palm, or Google Android, you can download the free Stitcher and listen to TWiV anywhere, streaming. So we thank Stitcher for doing that. We're also part of sciencepodcasters.org, promednetwork.com, and microbeworld.org, which are websites where you can find high-quality science podcasts in a wide variety of fields. David Bloom, Professor Bloom, thank you so much for joining us.
Well, thank you for having me. This is fun.
Great. Appreciate it. Professor...
Why are you laughing?
Professor McFadden, thank you for joining us.
My pleasure, totally.
It was great. And of course, my partner here, Rich Condit, always good to see you.
Well, thanks so much for visiting, Vincent. This has been just a great time. We had a really nice couple of days. You gave a great talk yesterday, and we've had a good time chatting and a wonderful time doing this TWiV.
No, I appreciate the invitation. I'm glad that we're able to do this. And thank the audience for joining us.
Yay, team.
1, 2, 3, 4, 5, 6, 7, 8, 9. 900 people in our audience. Fabulous. We have to do this more often. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.